Hello, my name's Rachel Jones. I'm a professor and an industrial hygienist, and I work at the University of California, Los Angeles Fielding School of Public Health. Today, I'm going to talk about the classes of chemical hazards. The learning objectives are to identify the primary types of health hazards associated with chemicals, explain key chemical physical principles and how they relate to exposures and health hazards, find relevant, credible sources of chemical hazard information, differentiate among information source types, and analyze the strengths and limitations of sources of information. A number of health hazards are associated with chemicals that are routinely encountered in workplaces. Here, I'm going to discuss the primary classes of health hazards associated with chemicals. It's important to anticipate and recognize the health hazards of chemicals so that action can be taken to prioritize strategies to prevent the most significant health effects. For example, one may want to develop a policy to eliminate or substitute carcinogens from the workplace owing to the severity of disease and lack of threshold for the effect. This requires identifications of chemicals anticipated, suspected, or known to be carcinogenic. Another reason is to ensure that exposure limits and industrial hygiene activities are relevant to the health effects. For example, a health effect triggered by a short-term exposure at a high concentration, like hydrogen sulfide, should be assessed through short-term exposure limits and short-term or real-time exposure monitoring. And finally, it's important to be able to communicate hazards to workers. Hazard communication is an integral component of occupational health and safety. Workers must understand the risks that they incur through their work so that they can make informed decisions about how to perform a job and whether to perform a job. A carcinogen is an agent with the capacity to cause cancer in humans. Cancer is a group of diseases involving abnormal cell growth with potential to invade or spread to other parts of the body. Abnormal cell growth that does not spread are benign tumors. A chemical is typically recognized as a carcinogen based on epidemiologic studies and/or studies in experimental animals, but can also be informed by in vitro experiments, assessments of structural activity, and other types of information. The consensus today is that carcinogens do not have a threshold below which there is no risk for cancer. This means that it's very important to limit exposures to carcinogens in the workplace as much as possible. Elimination or substitution of the carcinogen should be considered as the first option. The figure on this slide is a symbol in the globally harmonized system of classification and labeling of chemicals, also known as GHS. This symbol denotes a chemical that causes cancer or systemic health effects. The figure is a diamond shape with red edging and the silhouette of a person with a star emanating from their chest to represent damage inside the body. This symbol is used to reflect a variety of systemic health outcomes, not just cancer. But it is accompanied by more specific hazard statements and signal words. For carcinogens, the hazard statements and signal words are may cause cancer with the signal word danger, or if the chemical is suspected of causing cancer with the signal word warning. The assessment of carcinogenicity of a chemical will generally consider all of the available evidence. Typically, evidence from well-designed epidemiologic studies is given the greatest weight because these studies involve humans that have been exposed to the agent and experienced the cancer of interest. It is common that the epidemiologic studies involve studies of workers because chemical exposures in workplaces are generally higher than in other settings, and there is more likely to be information about the magnitude of those exposures. Studies in experimental animals is another type of evidence that can be used to evaluate the carcinogenicity of a chemical. In this type of study, animals are exposed to the chemical and then they are observed for the development of the cancer or other biological changes that are relevant to carcinogenicity. Biological mechanisms are the processes by which the chemical causes cancer. Information about these processes may be obtained from experimental animals, in vitro test systems like tissue culture, or from studies in humans. There are a number of organizations that evaluate and classify carcinogens, including the International Agency for Research on Cancer, also known as IARC. Another organization is the American Conference for Governmental Industrial Hygienists, known as the ACGIH, and the Occupational Safety and Health Administration, known as OSHA, also classify carcinogens. Each of these organizations have slightly different schemes, which we will review in a moment, but they all use a strength of evidence approach to evaluate data about chemical carcinogenicity. Here, I will focus on the degree of evidence system that's used by IARC and which is described in detail in the preamble to IARC monographs. As noted previously, the type of evidence that is considered are drawn from epidemiologic studies of humans, studies of experimental animals, and in vitro studies or other sources of data about biological mechanisms. Evidence of carcinogenicity is described as sufficient, limited, or as inadequate to interpret, showing the presence or absence of a carcinogenic effect. Sufficient evidence of cancer in humans or experimental animals means that a causal association has been established. That is, a positive relationship is observed between the exposure to the chemical and the increased incidence of cancer in well-designed studies. In animals, this association must be observed in 2 or more species, 2 or more independent studies of one species, or in both sexes of a single species. For mechanistic evidence, sufficient evidence means that results from several different experimental systems are consistent and coherent, which typically require a substantial number of studies on a range of endpoints in 1 or more species. Limited evidence of carcinogenicity generally means that the data are suggestive, but there are limitations to the studies or inconsistent results that limit confidence. More specifically, limited evidence in epidemiologic studies mean that a causal association is credible, but that chance, bias, or confounding could not be ruled out as a possible explanation of the result. In experimental animals, limited evidence could mean that not enough species or independent experiments performed. And in mechanistic studies, if the number of endpoints was limited or there were unexplained differences in results from studies of similar design, then the evidence could be considered limited. Inadequate evidence of carcinogenicity in humans, experimental animals, and mechanistic evidence means a couple of things. 1, that limited or no data are available, or 2, that available studies are of insufficient quality, consistency, or statistical power to permit a conclusion regarding the presence or absence of a causal association. It's important to realize that inadequate evidence of carcinogenicity is different than evidence that suggests a chemical is not carcinogenic. Evidence suggesting lack of carcinogenicity means that several adequate studies were available that are consistent and do not show a positive association between exposure and the studied cancer at any observed level of exposure. It is important to realize that the criteria used to determine whether a chemical is a carcinogen has a very high bar for evidence. That means that many chemicals may be classified as having limited evidence or being suspected carcinogens, but it can be difficult to demonstrate that they are confirmed carcinogens. As mentioned previously, there are several organizations that classify carcinogens. Such classifications are undertaken to provide authoritative determinations of carcinogenicity because it can be difficult for individuals to compile and assess all of the evidence. Carcinogenicity classifications are communicated to the users of chemicals on safety data sheets and in other resources about chemical hazards, such as the NIOSH Pocket Guide to Chemical Hazards. It is important to identify carcinogens because it takes many years for cancers to develop, and cancer can be a severe, often fatal disease. As a result, occupational health and safety professionals should prioritize eliminating carcinogens from the workplace or reducing exposures to carcinogens as much as feasible if they cannot be eliminated from the workplace. The International Agency for Research on Cancer, known as IARC, has 4 groups in their carcinogenicity classification. Group 1, carcinogenic to humans, is applied when there is sufficient evidence of carcinogenicity in humans, such as when there is strong evidence in exposed humans that the agent exhibits key characteristics of carcinogens and sufficient evidence of carcinogenicity in experimental animals. Group 2A, probably carcinogenic to humans, is applied when 2 of the following determinations have been made. Limited evidence of carcinogenicity in humans, sufficient evidence of carcinogenicity in experimental animals, or strong evidence that the agent exhibits key characteristics of a carcinogen. However, this classification can arise from some other evaluations of the evidence base. Group 2B, possibly carcinogenic to humans, is applied when one of the following determinations have been made. There is limited evidence of carcinogenicity in humans, sufficient evidence in experimental animals, or strong evidence that the agent exhibits key characteristics of carcinogens. There is no requirement for strong mechanistic evidence in exposed humans or in human cells or tissue. Group 3, not classifiable as to its carcinogenicity in humans, is applied when the agent doesn't fall within any other group and includes the determination that the mechanism of carcinogenicity in animals does not operate in humans. These classifications are described more fully in the preamble to the IARC monographs, which was last updated in 2019. A link to this document is provided in the suggested readings. The American Conference of Governmental Industrial Hygienists, known as ACGIH, uses 5 groups in their carcinogenicity classification. A1 is a confirmed human carcinogen. The agent is carcinogenic to humans based on the weight of evidence from epidemiologic studies. Group A2, suspected human carcinogen, is applied when there is limited evidence of carcinogenicity in humans and sufficient evidence in experimental animals that is supported by mechanistic evidence of key characteristics of carcinogens that are relevant to humans. A3, confirmed animal carcinogen with unknown relevance to humans, means that there is available experimental evidence in animals that suggests mechanisms and or dosimetry that the agent is unlikely to cause cancer in humans except under improbable routes or levels of exposure. A4, not classifiable as a human carcinogen, is used for agents that cause concern that they could be carcinogenic for humans, but which cannot be assessed conclusively because of a lack of human data, and the in vitro or experimental animal studies do not provide mechanistic evidence of key characteristics of carcinogenicity sufficient to classify the agent into another category. Category A5, not suspected as a human carcinogen, is reserved for an agent that is not suspected to be a carcinogen on the basis of properly conducted epidemiologic studies or evidence suggesting a lack of carcinogenicity in experimental animals that is supported by mechanistic data demonstrating a lack of the key characteristics of carcinogenicity. The ACGIH explains these classifications further, along with other health classifications, in their booklet TLVs and BEIs, which is published annually. The book can be purchased directly from the ACGIH. Of note, NIOSH, the National Institute for Occupational Safety and Health, no longer classifies carcinogens or uses the term recommended exposure limit in reference to carcinogens because the organization recognizes that there is no safe level of exposure to a carcinogen, and work exposure should be reduced as much as possible through elimination, substitution, or engineering controls. For more information about the NIOSH policy, please see NIOSH Publication 2017-100. OSHA has a regulation titled Identification, Classification, and Regulation of Potential Occupational Carcinogens, which is detailed in 29 Code of Federal Regulation, Section 1990. This defines a 4-step process that the organization uses. First, the Secretary of OSHA prepares a candidate list of potential carcinogens. In preparing this list, the Secretary is to consider data submitted to the organization by the public, specific government officials and reports, IARC monographs, and any other relevant data. A potential occupational carcinogen is any substance or mixture that causes an increased incidence of benign or malignant neoplasms, or a substantial decrease in the latency period between exposure and the onset of neoplasms in humans or in one or more experimental mammalian species as the result of any oral, respiratory, or dermal exposure, or any other exposure which results in the induction of tumors at a site other than the site of administration. This includes any substance which is metabolized into one or more potential occupational carcinogens by mammals. After this candidate list is developed, it is prioritized. Factors of the prioritization include the number of workers exposed in the United States, the levels of human exposure, the extent to which regulatory action could reduce the risk of cancer or other hazards, structural similarity of the compound to other carcinogens, the availability of substitutes that are of lower health risk to workers, and actions that are planned or being taken by other agencies that could impact exposures to this chemical. After prioritization, the list is published annually in the Federal Register. Rulemaking may then be initiated for potential carcinogens. At this time, there are fewer than 30 agents that are regulated by OSHA as carcinogens or potential carcinogens. These are generally the most widely recognized carcinogens, such as asbestos, benzene, and methylene chloride. Mutagens are chemicals and other agents that cause mutations or alteration in the base pairs of DNA. The impact of mutations depends upon the type of change in the DNA and the type of cell in which that change or mutation occurs. Mutations may involve substitutions of bases in the DNA sequence, deletion of bases, or insertion of additional base pairs in the DNA. Mutations can occur in somatic cells. These mutations can have varied effects, including some of which do not have notable manifestation. When these mutations result in alteration of cell division, it can lead to the formation of cancerous cells, that is, cells that reproduce at abnormal rates. Mutations that occur in reproductive cells or gametes are heritable and can be transmitted to the progeny, either in the somatic or reproductive cells. Reproductive toxicity is the event of biologically adverse effects on the reproductive system of females or males. This toxicity can alter female and male reproductive organs, the endocrine system, and pregnancy outcomes. Effects of this toxicity can manifest in many different ways, including the timing of the onset of puberty, menstrual cycle abnormality, sexual behavior, infertility, altered sex ratio in offspring, spontaneous abortion, and many others. For more information about the types of reproductive toxicity, the EPA's 1966 Guides for Reproductive Toxicity Risk Assessment provides some background information and describes how some of these outcomes are measured in experimental animal studies. Developmental toxicity involves adverse effects on the developing organism that results from parental exposure that may occur prior to conception, during prenatal development, or postnatally. Developmental toxicity is one type of reproductive toxicity. A teratogen is an agent that can cause a birth defect and is a type of developmental toxicity. Reproductive toxicity, developmental toxicity, and teratogenicity are not necessarily distinct groups. Multiple types of toxicities in these categories can be caused by a single chemical. For example, 2-methoxyethanol, which is also known as methylcelisolve, and ethylene glycol monomethyl ether, or EGME, is a reproductive toxicant because it causes testicular atrophy, decreased sperm motility, and increased incidence of abnormal sperm. The chemical is also a developmental toxicant and a teratogen because it causes toxicity to the embryo that results in developmental effects in the offspring, including reduced fetal weight, fetal loss, and skeletal defects. For this chemical, most of the data about adverse health effects is from studies in experimental animals, and for these organ systems, the biological processes are analogous in these animal species to that in humans. Epidemiologic studies for these outcomes can be difficult as the number of exposed workers is small and distributed across many workplaces, and few workers are attempting to conceive or are pregnant at any specific time. Thus, it can be very difficult to detect these types of outcomes among groups of workers. Chemicals can affect a variety of organs and organ systems in the body, not just the reproductive system. I'm going to talk briefly about the different organ systems and identify a couple of examples of adverse health effects that are known to occur among workers. The goal here is not to memorize all of the organ systems and health effects, but to simply appreciate the wide variety of ways that chemicals can negatively impact the human body. The integumentary system, or the skin, includes all components of the skin: including the epidermis, dermis, hypodermis, glands, hair, and other components. The picture on the right shows the complexity of the skin. A common adverse health effect affecting this system is contact dermatitis, which is an irritant-appearing rash that can occur on any point on the body where it has contacted an irritant. The hematopoietic system includes the organs and tissues associated with the production of cellular blood components, including the bone marrow. Some chemicals affect the ability of the body to produce blood components. An example is benzene that can affect the bone marrow to cause a decrease in red blood cells, leading to anemia, among other adverse effects, including leukemia. Other chemicals affect the circulation or oxygenation of blood. An example is carbon monoxide. Carbon monoxide binds to the heme in the blood more strongly than oxygen and can cause cyanosis. Cyanosis is the event that the lips or fingers turn blue owing to low oxygen levels in the blood. The digestive system includes the pathway from the mouth to the anus and the associated glands and organs, including the liver. Jaundice is the event that skin takes on a yellow color, which comes from bilirubin and indicates a problem with the liver. Jaundice may be caused by viral infections of the liver, such as hepatitis, which can be acquired through workplace exposures. The urinary system includes the kidney, bladder, ureters, and urethra, which filter blood to excrete liquid waste. Mercury is a chemical that causes tubular dysfunction in the kidney, which is detected by increased levels of urinary proteins. The nervous system includes the central nervous system, comprised of the brain and spinal cord, and the peripheral nervous system, which is made up of the nerves that branch from the spinal cord. Exposure to many organic solvents can cause depression of the central nervous system that can manifest in symptoms like drowsiness, dizziness, incoordination, and reduced heart rate. Exposure to a combination of N-hexane and acetone can affect the peripheral nervous system to cause peripheral neuropathy, which is weakness, pain, or numbness in the hands and feet. The ocular system includes the eye and central visual system, which is the cornea, lens, and fluids. Some organic solvents, including styrene, can cause a loss of color vision. Conjunctivitis is the inflammation of the membrane that covers the front of the eye and inside of the eyelids, which can be caused by bacteria and viruses, physical irritants, and other agents. The auditory system is the sensory system for the sense of hearing. Some organic solvents, including styrene, can cause hearing loss. Finally, the respiratory system is the tissues and organs that help with breathing. There are many lung diseases caused by or made worse by exposures to chemicals. Cotton dust, coal dust, asbestos, and other agents cause lung scarring. Isocyanates can cause work-related asthma. Some chemicals can cause an allergic response. A type 1 response is an IgE-mediated response, which occurs when exposures to the antigen stimulates mast cells to produce histamines, prostaglandin, and other biological chemicals. An example of a type 1 response are seasonal allergies. A type 4 response involves sensitization. When an individual is sensitized to the antigen, their body is primed for response, and they may experience a severe reaction at their next exposure, even when their exposure to the antigen is low. An example is beryllium, where exposure can lead to sensitivity in the short term or the long term, and then can progress to chronic beryllium disease. Corrosion is a direct effect of chemicals at the point of contact and involves the destruction of tissue. Corrosion most commonly affects the skin, eye, or respiratory tract. For skin, this means visible necrosis through the epidermis and into the dermis, such as ulcers. For the eye, this means serious or irreversible effects to the cornea, iris, or conjunctiva. Hydrogen fluoride is an agent that causes particularly severe tissue destruction. The figure on the right in this slide is a symbol from the GHS system used for corrosive materials. The figure is a diamond shape with red edging with two images inside. One image shows a liquid being poured from a test tube onto a rectangular piece of material with wavy lines coming up and showing the loss of material where it has been contacted by the liquid. The other image is similar but shows a hand instead of a piece of material, where damage to the hand is visible after contact. It is important to recognize that dermal exposures to chemicals may lead to the chemical being taken into the body to cause systemic effects without causing corrosion to the skin or to irritation to the skin. But some chemicals are capable of causing corrosion or irritation. Irritation is different from corrosion because it involves reversible damage to the tissue, and this includes drying of the skin. For skin, the redness or swelling that manifests with irritation reverses after exposure ceases. Drying is the event that chemicals like soaps, solvents, and water remove natural oils from the skin, causing it to become very dry. Irritation to the eye can involve conjunctival redness or swelling, changes in corneal opacity, and irritation that reverses after exposure ceases. The figure on the right is the symbol in the GHS system used for irritants and other agents that cause acute toxicity. The figure is a diamond shape with red edging, and inside is a black exclamation point. Acute irritation in the respiratory tract manifests differently in different locations. An important factor of where the irritation manifests is the water solubility of the chemical. More water-soluble agents are likely to irritate the upper respiratory tract more than the lower respiratory tract. Irritation in the nasal and paranasal sinus often manifests as congestion. Acid mists may cause such irritation. In the throat, inflammation from acute irritation may manifest as a dry cough. Sulfur dioxide or fluorine can result in cough. Inflammation in the larynx manifests as hoarseness in the voice, and if severe, as shortness of breath or glottal edema. Irritation in the middle of the respiratory tract manifests typically as bronchospasm, but can result in asthma. Many agents cause asthma, including isocyanates, high molecular weight compounds from animals, plants, metals, and some wood dust. Irritation in the deep respiratory tract manifests as pulmonary edema and pneumonitis. Pulmonary edema arises from the toxic action to capillary walls and can result from exposure to ozone and nitrogen oxides at high levels. Pneumonitis is an inflammation of the lung parenchyma. Asphyxiation is the state or process of being deprived of oxygen. This can result in unconsciousness or death. There are two main types of asphyxiants. First, inert or simple asphyxiants. These act by displacing oxygen in the atmosphere. An unfortunately common context for exposure to inert asphyxiants is in confined spaces where a gas or vapor has displaced oxygen. A second type are chemical or toxic asphyxiants. These chemicals act by interfering with the transport, delivery, or utilization of oxygen in the body. Examples include carbon monoxide, which binds to hemoglobin to form carboxyhemoglobin and interferes with oxygen transports to tissues. Another example is hydrogen sulfide, which interferes with oxidative enzymes to cause tissue hypoxia. And the third example is hydrogen cyanide, which impacts the enzyme cytochrome oxidase that is necessary for tissue respiration. Here we have some suggested readings, most of which have been mentioned at some point during this presentation.